everybody, this is video number 5. This one is going to be, well, this is in the DNA replication, transcription, and translation series, and we're finally getting to translation here. So this is going to be all about translation. Just a little bit of word of warning, this one could be long because translation is a fairly complex process, and we have to explain some things first before we actually get into translation. Just a quick reminder here that the goal here is to make a protein, and a protein is a chain of amino acids called a polypeptide that's folded into a very exact shape. And in order to get that chain of amino acids, this is an amino acid, this is an amino acid, this is an amino acid. In order to get them in the exact order they have to go, because if you get one wrong, you've goofed it up. There are 20 amino acids. Think of them as kind of like, I don't know, like you guys remember when people used to make those necklaces and you had these beads that had letters on them and you could spell out somebody's name or something on those necklaces? Well, these are kind of like that, and if you got one letter wrong, then you misspelled whatever you're trying to spell. Well, the same thing here. These things go in a very specific order, and if you get one out of order, then you don't make the right protein, and that's bad, because if the protein you're trying to make is insulin or hemoglobin, and if you don't make insulin, then you end up having diabetes. So this is the process here, and that's what we mean by the genetic code. The gene, the order of DNA up here is the genetic code. We copy that onto the RNA, and we use that copy to put these amino acids in the exact order through this process called translation. How do we put amino acids together? Well, sorry about that, back up here. Okay, an amino acid is all the exact same shape. It's got this amino group here, this carboxyl group here, and this group in the middle, and then there's what they call the R. Now the R group is what's different about every amino acid. There are 20, and they all have the same structure here, but the R is what's different. Here is alanine. That's got a the amino group over here, and then it's got this structure here. But look at serine, same thing there, except this red region is slightly different. And each amino acid is slightly different, but we stick them together using what's called a peptide bond. Now, making that peptide bond between these is what happens during translation, and we put them in order next to each other, and then we stick them together. It's kind of like making a train car. Okay, like a train. You put a bunch of cars together and you hook them together. That's exactly what we're doing here, because remember that we're making a polymer out of a bunch of monomers. Let me remind you, this is a monomer, and this is a monomer, and this is a monomer, and when we put monomers together in this order, we get a polymer, and a protein is a polymer. Now in this case, the polymer has to be very specific how we put it together, because we have 20 different amino acids and we use a whole bunch of them over and over again, but we got to put them together in a specific order. So putting them together in order is kind of the whole ever-loving point. How do we get them together, go together in that order? So here's a little bit longer look at this polypeptide chain, and each one of these little balls is an amino acid, and the order that they are, here's all the amino acids, and the order that they are is important, because that's what makes the protein. If you get one of these wrong, you don't make the right protein, and you have to make the right protein or else it could have serious effects. If this is insulin and you get one of these, just one of these wrong, then you don't make insulin any longer, and you have a real problem. You're diabetic. So we have to get these amino acids in the exact order. How do we do that? Well, we've got the order coming to us because it's found on DNA. That's what the gene is, and we've got that copied over to the RNA. But how do we take the RNA and use it to get these amino acids in the exact order? And this process is again called translation. Now here's the thing. On the RNA, there are all of these nucleotides are grouped into threes. Okay, so here's adenine, uracil, guanine, and they're grouped into threes. We call that a codon, C-O-D-O-N, and they're always in threes. And these codons are what the genetic code is actually grouped into these groups of threes. So the RNA, the messenger RNA, when it comes out, is going to be some multiple of 3. Okay, it has to be. So it has to be like 3,000 nucleotides long. It can't be 3,001 because that's not a multiple of 3. So it comes into groups of 3s, and these 3 are called codons. Now the information then, helps us to put the proteins in order, because watch this. Well, let's talk about how to read codons, because each codon here actually specifies for an amino acid. I'm pointing up here because I want you to start thinking, if I have this codon here on the messenger RNA, then up top there's going to be an amino acid up here that goes with these three codons. Three codons equals one amino acid. Here's another three codons, one amino acid. Here's another three codons, one amino acid. So this tells me what order to put them in. Okay, so I have AUG, and this is called a mandala, and if you like it, you can read it. I don't particularly care for it. Okay, but it does work. The first letter of whatever you're looking for, like let's look at AUG. The first letter is found here in the middle, A, so that's going to be here. Then we go right here, then it's U, so there's U right there. So A, U, and then G, and that tells us the amino acid. In this case, it's methionine, and all genes start with this AUG codon. It's always AUG. I always remember it as AU means gold and G is gold, so AUG for gold, but that's how it starts. Um, this is every gene starts with this because those um, this is how those enzymes know where to start copying the DNA. Is it finds methionine, and when it finds methionine, it says, okay, we start here and then we keep going. So let's look. The next codon is AAC. So we've already got methionine lined up, and I'm going to show you this again in a minute, but we've already got methionine lined up. That is an amino acid. It's sitting at the top ready to go. Now we go to, what was it? AAC. So now we go to A, and then C. So the next amino acid is asparagine. So now I've got a methionine next to an asparagine, and then the next, what was the other one? UCU. So now we go up here. Here's U, and then there's C, and then there's U, and that's serine. So we pop the serine in next. So now we have already three amino acids in order: methionine, asparagine, and serine. And pardon me, that could be this: methionine here, asparagine, and then serine. Okay, but we just keep going, and there's just a continuous number of codons. Could be thousands long. However many amino acids you see, you multiply that times 3, and I'm not going to count these, but for every amino acid you see, you have 3 nucleotides. That's how long the gene is, right? So this mandala tells us what goes where. Now, I don't like the mandala. I kind of like the square better. Okay, and the square works the same way. The first one was AUG, so we have the first letter was A, so then the second letter was U right there, and then the G, that gives us methionine. Okay, and I just know how to read those. I don't know which one you like, but you are going to have to use these, so you should pick one. You might like the mandala. It may work better. I don't know, but I like the square here. But either way, you can have whichever one you want. But let's do these 6 different codons right here. UUA, which would be UUA, that means leucine, right? So there's a leucine. And then UUG, which is UUG. Wait a minute, that's also leucine. Can you do that? Yes, you can. In fact, if you'll notice, a lot of these have different codons, but they mean the same thing. There are 20 amino acids. Some of these are repeated. For instance, here is, let's see. There's leucine there. As you see, there's another leucine there. There's 4 codons here for leucine. There's 2 codons. There's at least 6 codons for leucine that I see. So just because you have, there are 64 codons and only 20 amino acids, so we got to double up some. Now some of them, like methionine, only has 1 code, AUG. That's how you get methionine. And here's one called tryptophan, and I think you only get tryptophan one way, and that is UGG. That gives you tryptophan. There's a couple of stops. Remember, methionine is start, and there's 3 stops. So any one of these could tell the thing to stop copying, right? But other than that, there's 4 there for glycine. Is there glycine anywhere else? So there's 4 codons that tell you to put a glycine. So just because I got different codons doesn't necessarily mean there's going to be a different amino acid at the top. UUA and UUG both mean leucine. So now I've got 2 leucines, and then after that is a CUU. So here's a C, a U, and then a U, which makes another leucine. My goodness, there's a third leucine. CUC. Oh, hey, finally. Nope, sorry, it's another leucine. CUA, that's another leucine. CUG, yeah, all these make leucine. So Yeah, if I'd have just read that, that's what it says. So if this were my code, it would be leucine, 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 leucine. Isn't that crazy? Um, but if you look at tryptophan, only UGG makes tryptophan. There's no other codon for tryptophan. So something to notice about these codons is that it's not a one for one. There are 64 codons and only 20 amino acids, so a lot of codons are going to go for the same amino acid. And then, of course, we've already talked about the start and stop codons. MET is start, and then there's three stops: UAA, UAG, and UGA. So any gene starts with an AUG and ends with either UAA, UAG, or UGA. Has to. That's the only way that um, RNA polymerase knows where to start and stop, right? So then we finally talk about translation, and the idea is pretty straightforward. I have the information on my messenger RNA. I need to turn it into a sequence of amino acids. This is done outside of the nucleus. Okay, transcription happens in the nucleus, translation happens outside of the nucleus. And here comes my messenger RNA that I made. It's coming out through these holes in the nuclear envelope or the nuclear membrane called nuclear pores. It comes out through these pores. It's pretty long, as you can see. But look, there's 1, 2, 3, it's a codon. 1, 2, 3, that's a codon. 1, 2, 3, that's a codon. 1, 2, 3, that's a codon. So it's nothing but a series of three codons. Keep that in mind, okay? Now what happens is that um, as it moves through the ribosome, it kind of, I don't know how to describe it. One end just kind of sticks into the ribosome, and the ribosome kind of moves along it and reads it, okay? And it reads, so there is a start codon, AUG, that's start, okay? Now all three, by the way, let's stop for a minute. All three types of RNA, there are a bunch of types, but the three that we study are in this picture. This long strip is called a messenger RNA. That's it right there. This long strip is called a messenger RNA. This big blob structure right here is called an rRNA. It's also a ribosome. So we studied ribosomes back when we studied cell structure. Well, this is a big ribosome. Okay, that's also the rRNA. And then finally, there is a third type of RNA called tRNA. T stands for transfer. This structure here is called a transfer RNA. Now, it's interesting because on the top it has an amino acid, and there's only one kind of amino acid that can go on there. That's what the mandala tells you. Okay, if you have the codon, then you know what ultimately is going to be up there, right? So this is called the codon. We're going to talk about this right here. Here's your codon, AAA. That is entirely possible. Let's go look and make sure. AAA is lysine, right? So here we go. We have the codon here, AAA, that's for lysine. On the bottom of the tRNA is the kind of the complementary codon. We call it the anticodon. It's UUU because what does A always bond with in RNA? A normally bonds with T in DNA, but we're talking about RNA, so these are all uracil. So this is UUU. This, if you think about it, this is the only place it can go. It can't go anywhere else. That's how we know that tRNA goes there. And whenever you have a tRNA that has UUU on the bottom, there's always going to be a lysine on the top, every time. So that thing is going to stick right there. The ribosome grabs that tRNA and it reads, well, okay, let me rephrase this. The ribosome, as it moves along, it reads this codon and says, oh, I've got an AAA. All right, I need a UUU. So it grabs this UUU and sticks it here. And it's going to keep reading it. Okay. Now, it's already happened a little bit, so let's start back here. We have AUG. The anticodon for that is UAC. Okay. The ribosome reads AUG. It says, oh, I've got AUG. I need to find the tRNA that has UAC at the bottom of it. Grabs it, puts it on there. There's the methionine at the top. Okay. Now then, the ribosome snaps that methionine off and sends this tRNA without the amino acid. It sends it away. Now this tRNA will go find another amino acid, another methionine, and stick on top of it. But that just happened somewhere else. Then the ribosome says, okay, now I have a UUC codon. The anticodon is AAG, so it grabs the tRNA that has the AAG at the bottom and puts it there. Bink. Then right at the top, the only thing that's going to be on this tRNA is phenylalanine. So it sticks this methionine to this phenylalanine, pops it loose, bink, and sends this tRNA off again, and it has to go find another phenylalanine and come back. Then it reads the AAA and it says, okay, I need a UUU. So it grabs the tRNA with UUU on it and bink, sticks it there. It takes the methionine and the phenylalanine that it's already popped loose from these tRNAs and sticks it onto this lysine, bink, breaks that loose, bink, throws this tRNA out, says go find another lysine, and it keeps doing that all the way down. And as it goes all the way down, and you can read this if you want. I guess I should slowly. I'm telling you all this. Sorry. But it keeps going down in order until it gets to the stop. And that's when it's at the end. And then it's made its gene. So each one of these little guys right here was once attached to a tRNA. And then the ribosome read the mRNA, found the right tRNA, and put that right there. And then put the next one. And then it put the next one. And it moved it all the way down. You can imagine that the mRNA is kind of alongside of this, and it puts these in a specific order, and it can't screw it up, right? And I say can't, it can, but it normally doesn't screw it up because it's just reading what's on the mRNA, which was a copy of the DNA, and it turns it into this chain of amino acids, and it makes this exact chain. And the difference between proteins is just that these amino acids are in a different order, and they're longer. You know, some polypeptide chains are long, some are short. Now I'm going to put a video in here for you to watch to help you review this, but that's a tough one, okay? It's kind of tough to follow what's going on here. So there's a really good video I'm going to include that kind of talks to you about all this transcription and translation. But pay very close attention to how these amino acids get in order, okay? And remember, if I get one amino acid out of order, boop, it's goofed up. Pardon me. And also, here's the difference. If you have dark skin versus light skin, it's because you have proteins that make you have dark skin, whereas people with light skin don't have as many proteins. It's called melanin. Melanin is a long protein. If you have a bunch of it, because your genes say make a bunch of it, then you have darker skin. If you're poor, like me, I wish I had darker skin because I go out in the sun and I get crispy crittered because I don't make as much melanin, right? And so that's all. That's it. That's the difference between, races is, the skin color. It's just, do you have a bunch of melanin in there or not? That's all it is. Are your eyes different colored? Same thing. There are proteins that color your eyes. There are proteins that color your hair. There are proteins that shape your nose. There are proteins that shape your hands. There are proteins that make you smart in math. There are proteins that make you smart in music. There are proteins that make it where you can run faster. Every single thing about you comes down to proteins, okay? Whether you have them or you don't. And if you have proteins, if you have certain proteins, you are dark skinned. If you don't have them, you're light skinned. If you have them, you have brown eyes. If you don't have them, you have light colored eyes. If you have this protein, you're probably pretty good at music and math. If you have this protein, you're probably good at painting. If you have this protein, you may be good at running. If you have this protein, you may be good at, I don't know, singing. That's just the way it works, okay? And that's how DNA works. That's the whole idea behind genetics. You inherited all of those DNA from your parents, and then we take that DNA and we make this protein out of it. And that's why you kind of look like your parents, because you got the same information that's making their nose that shape is the same information that's making your nose that shape. I want you to chew on that for a while because that's kind of a big deal. And I want you to know something. This right here can change the world. It is going to change the world. In fact, it's going to be kind of scary. What we can do by learning how to manipulate these genes or add and subtract to these genes, because we're doing that right now, being able to do all that is going to change everything. It'll cure global warming. It'll cure diseases. It'll cure hunger. It can also cause a whole new set of problems. So it's something you really should learn about because it's kind of scary. Okay, that didn't take as long as I thought it would. I think mostly because I want you to watch the video that I attached with this. So there's going to be this video and another video that I want you to watch. Okay, it explains it very well. Watch it a few times and then take the check yourself quiz. And we're going to have one more video in this series, and then that'll be the end of it. Okay, this ends video 5. Watch the other video and then go take the Start Here quiz. Thank you.